Hi, good morning. My name is Sid Bhoopathy. I work at a company called Absorption Systems. And I would like to thank the organizers for the kind invitation to attend and present at this event, and also for the organizing committee to be so accommodating and allow me to actually combine my two presentations so that I can put together a more cohesive story. So today I'll be talking about how do we conduct performance evaluation for what is known as complex drug products, and some of the insights that we have developed as we have conducted research on this for a few years now. So before we get into the specifics, I just want to get a few definitions in place so that the room is thinking about this the same way. So complex drug products can either be complex active ingredients, could be polymers, natural source products, and so on. They could be drug device combinations. They could be complex formulations that are made up of liposomes or other materials such as iron colloids. Or they could be a non-oral route of administration. And today's focus will be on the complex route of administration, which includes both the non-oral and the locally acting drug products. So many times during development, as you've established your first prototype and as you progress, one of the key objectives is to maintain therapeutic equivalence. And the definition of therapeutic equivalence is that you have the same clinical efficacy and safety profiles when administered to patients under conditions specified in the labeling. So essentially the ability to substitute, to have interchangeability without any adjustment in dose or additional monitoring. To establish therapeutic equivalence, it is important that you have bioequivalence. And to establish bioequivalence, it is desirable to begin with what are known as pharmaceutical equivalence, where your test is the same API as the reference. So when you have pharmaceutical equivalence whose rate and extent of absorption are not statistically different, then it is possible to say, well, as a surrogate, you probably have therapeutic equivalence. There are many approaches to determining bioequivalence. Most of you, of course, are familiar with this. There is the human pharmacokinetic approach. You could take the pharmacodynamic approach, clinical comparison, and there's also the possibility of using in vitro techniques, and we will talk about that some more as I elaborate on the multi-platform testing. So again, the focus of today's talk is complex drug products, and within that, the locally administered, locally acting, and the non-oral drug products. So to establish the challenge, when you have something that is topical and locally acting, and if the question was like, how do we establish bioequivalence? The current standard is essentially to perform a clinical endpoint study. Now these studies are difficult to perform, they're arduous. And the reason is that these tests are inherently very, very insensitive. So a typical design would be you have a 3-arm comparative trial where you have the reference, you have your test, and then you have the placebo. And not only do we need to demonstrate that the reference and the test are of course better than the placebo, but also that the reference and the test are statistically similar. And essentially this is a dichotomous outcome where you have either pass or fail depending on your confidence interval. So these tests have high variability, they have low sensitivity to formulation changes, and it also requires a lot of unnecessary human testing, and therefore it has become a barrier to product development. A clinical endpoint study essentially applies to any therapeutic because all of them have a basis for a clinical endpoint when they progress through their initial development. But there are also other possibilities, other mechanisms for establishing bioequivalence for topical products depending on product-specific factors or certain mechanisms of action. And these would be studies such as the vasoconstriction study, which applies to corticosteroids, certain types of tape stripping studies, which were introduced in the late 90s, but that guidance has been pulled now. and also the possibility of using dialysis probes where you're measuring the concentration of the drug substance in the dermis. But even these alternate studies are not easy to perform. They're expensive. There are few in vitro established pathways to obtain waivers or exemptions from the in vivo path. And there's also a lot of complexity when you're working with semi-solid formulations. Complexity which pertains to its application, complexity that pertains to the release of drug for the formulation, complexity that applies to the physiology where you actually place the drug product, because there are several pathways through which this drug product can enter into the systemic circulation. Therefore, there has been this initiative by regulatory science, which is essentially looking at what are these alternative methodologies that can be looked at for assessing, for establishing equivalence of these complex drug products. And the crux of this, the central focus of this, is through the use of biopharmaceutics, which is a study of the chemical and physical properties of drugs and the biological effects that they produce. And the hope is that with a sound understanding of biopharmaceutics, we have the ability to predict bioequivalence parameters and infer therapeutic equivalence without always having to perform clinical studies. And there is precedent for this. This has been done before in the non-complex drug product space. And I want to illustrate this to you before we talk about how we could maybe take this challenge on in the complex drug product space. So the graph here illustrates what happens with typical oral dosage form performance. So you ingest, the drug product disintegrates, it dissolves, and then it is absorbed through the gut wall, through the liver, onto systemic circulation, reaches the plasma, and then the site of action essentially succeeds the central compartment, the plasma, before activity is elicited. So in this situation, there are tests. There's a scientific framework that is well established, known as the BCS, that applies to certain drug products, more specifically immediate release drug products, where if the parameters that are known to impact, that are known to cause differences in rate and extent of drug absorption, cease to be rate limiting. And these parameters would be parameters such as solubility, permeability, and dissolution of the drug product. If these cease to be rate limiting, where essentially in theory you're administering a solution to the relevant intestinal absorptive surfaces, then a biowaiver is possible, as in you do not need to perform a human pharmacokinetic study to demonstrate equivalence. The biopharmaceutics of the drug substance combined with the rapid dissolution from the product makes bioequivalence self-evident. And this has been put in practice for many, many years now. Applies primarily to BCS Class 1 drug products, can also apply to BCS Class 3 depending on the jurisdiction. And essentially what this graph illustrates is the correlation between human absorption versus what you obtain using an in vitro model like the CACO2 cells. The x-axis, I do apologize, I cannot get the pointer to work.
Just push another button.
Other button? Okay, thank you. So that was easy. So in this illustration, the x-axis here is the in vitro surrogate. Data here specifically is from the CACO2 cell monolayer model. And the y-axis is human absorption. So this is data from maybe about 60 drug substances. Quadrant 1 is the true positives, where the human data correlates with the in vitro model. Quadrant 2 is where there is apparent conflict, but that's also because the human data is much more variable. This is true negatives, and this is where the in vitro is conservative. So what this essentially shows is that there's remarkable accuracy. There's about 85% correlation between in vivo and in vitro's ability to predict what can happen in the human situation. And this is the basis for a BCS-based biowaiver for immediate release solid oral dosage forms. But now when you look at the complex drug space, not as simple as the oral situation, because you have release from the product, and passage through the skin can be through more than one route. The site of action could be in the skin layers, which results in the effect, and the drug makes it to the bloodstream, again through multiple pathways, either directly without even reaching the site of action, or from the site of action as it is eliciting the effect. So what that does is it makes the central compartment, the blood compartment, less useful as a means of establishing equivalence. And therefore, the direction is more towards these clinical endpoint studies or other product-specific or mechanism-specific factors. So the goal in this presentation is to talk a lot about this idea of using weight of evidence, where recognizing the complexity of the formulation, recognizing the complexity of the pathway that this formulation has to take. Maybe it is not as simple as just putting together solubility, permeability with dissolution, but there might be methodologies that we could use where if you have a combination of approaches pointing in a certain direction, there could be more confidence that truly you have equivalence without having to always perform these clinical or endpoint studies. And building the weight of evidence begins with first showing that the product you're trying to develop, especially if your goal is equivalence, has similarity to the reference, that you have the same components as the reference, that you have the same components at the same concentration, and that you also have the same arrangement of matter, as in the microstructure between the formulations is similar. And this is easier said than done, and for which you require comprehensive testing that involves looking at physical attributes, making sure that your inactive ingredients do not influence its permeation, uniformity, homogeneity, impurity profile, and also the release from the formulation. So the direction we would like for the science to move towards is getting away from this reliance on always using human studies, all risks of inequivalence primarily managed by design of bioequivalence, to more of a balanced approach where yes, B is still critical because it complements what you learn in your early investigation, but that should not be the sole pathway, and an increased emphasis on pharmaceutical equivalence where quality by design is established through dosage form assessment, evaluating the strength of the formulation, and by product design and product performance. So what that means is a cluster of tests, and these tests are broadly known as performance evaluation, and they have several objectives. Performance evaluation can be used for dosage form assessment. So essentially you can screen and optimize what needs to go into the formulation. It can be used to study impact of excipients if you had to substitute because the Q1, Q2 is not possible. Or it can be used to select a lead or a prototype formulation, which could be your basis for a pilot or a pivotal clinical study if such is required. A well-designed in vitro technique or set of techniques can also be used to establish bioequivalence, but for that you have to show that your technique can actually discriminate when there is a difference in the formulation, and that also it can have parameters of reproducibility in a consistent manner. And then therapeutic equivalence, clinical endpoint studies, but what about the use of non-clinical disease models, and can they be used as surrogates, again, to build this weight of evidence type of approach? So some of the tools that exist for evaluating equivalence or performance of complex drug products, broadly under this label of multi-platform testing, a plethora of different tools. They can be a combination of in vitro, pharmacokinetic, and disease models or pharmacodynamic models. So tools within a bucket, increasing complexity as you go from synthetic membrane to cell monolayers through three-dimensional tissue equivalent and excised tissue, and also increasing complexity as you go from in vitro to pharmacokinetic to pharmacodynamic. The deliverables also of increasing prominence. In vitro, you could look at a variety of different variables. Feasibility, is this route even possible? You could optimize different variables such as pH and excipient composition. You could look at would repeated use cause inflammation? What about biochemical interactions such as uptake by transporters or metabolism? You could look at regional absorption in excised tissues. Pharmacokinetic is essentially the PK profile, the distribution, PBPK, it can be used to set up input parameters for PBPK. And pharmacodynamic is where you're looking at efficacy and IVIVC translatability to the human situation. So within performance testing, the next part of the presentation, essentially, I have case studies and specifics on how we have used this to derive some of these endpoints. You could use this for multiple locally acting products or non-oral routes of administration. I'll only focus on two. I'll begin with intraoral, just give you a sense of how some of this can be connected, and then talk in a little bit more detail about Ocular, where we have done much more extensive testing along the lines of the BCS framework to show that ocular drug products can also avail this pathway. So for intraoral application of multi-platform testing, in this situation, we are working on a drug product that is essentially administered orally. And this was for a maintenance regimen. So you use it for many, many years to arrest disease progression. What we're trying to develop was a lozenge for an indication that was not primary, but in this case, more as an acute relief of cough. And thought was, since you needed more rapid relief, is it possible to maybe administer the same drug substance instead of orally as a lozenge? And then using these types of tools to first answer the question, A, is it feasible? And then can you take it on to the translatable outcome of actually demonstrating that you have similar results in a human situation? So we used cell monolayers, we used buccal tissue, porcine buccal tissue, and then also preclinical PK. Started off by first understanding what are the basic permeation properties of the drug substance. The compound is moderately high permeability, right here on that scale that we had seen before, as in it has good intrinsic permeation parameters. It is also passive. And then the next question was, well, what happens in a tissue situation? Because cell monolayers don't necessarily sometimes mimic what happens when you have many more layers of tissue. And that's when we realized that it did not perform as well, the flux across to the other side, and it did better when we were to use some permeation enhancers such as menthol. So taking this knowledge forward, we performed a preclinical PK study. In this case, again, the control is where the drug substance is administered orally. And the idea is, with this lozenge, intraoral delivery, we are hoping for more rapid onset, so quicker Tmax, also a higher Cmax, and then more rapid elimination. This was the desired profile that we were pursuing. And it's a little tricky when you perform such studies in preclinical species, because the physiology is different, and sometimes you'll have to humanize these outcomes. So this was a dog pharmacokinetic study, and in dogs, the basal pH is higher than humans. So when you have higher pH, sometimes that can have an impact on solubility, it can have an impact on the tubular reabsorption. So there were many methodologies that we used to make the dog more human-like, control its salivation, make its urine pH more acidic, and also make its gastric pH more acidic, so that we could get that more similar to the human outcome. So with this, the Cmax, we were able to increase it. The Tmax reduced from almost like 45 minutes to 15 minutes, and also the half-life of the drug substance was reduced from almost 7.5 to about 4 hours, indicating that we were on the right track and that a lozenge was indeed possible, and currently this is in phase 2 clinical trials. Similar idea here, where the test in three-dimensional tissue models is assessed for excipient change. So two different formulations. The question is what happens with and without EDTA. So with EDTA, so here the load is either 2 milligram or 8 milligram. The first two rows are with EDTA, the second two rows are without EDTA. So the question being asked is, does EDTA have an impact on the mean flux? The data is normalized to the loading dose, and essentially what this demonstrates in the three-dimensional tissue model is that it does not make a difference. The EDTA is not in this particular situation acting as a permeation enhancer. Also corroborated through in vivo exposure, where with or without EDTA, there was no difference in the overall pharmacokinetic profile. So one thing to keep in mind when you consider these types of tools to either guide your formulation strategy or establish equivalence is the inherent variability. So going from KCO2 to more of a tissue model or recirculating perfusion, there is usually an increase in your percent CV. It is important to keep that in mind because that is how you would power up the study to ensure that the model has the ability to dose discriminate. Otherwise, you'll always get a result that demonstrates equivalence. Also, the dynamic range, usually better for an in vitro model, and then it reduces when you move towards progressively more complex models such as excised tissue or intestinal perfusion. Ocular delivery. For drug products in this space, the current guidelines for establishing equivalence are very challenging. And that's why there is a need to come up with alternate solutions. And again, we applied very similar concepts. The idea is, can you take some in vitro test systems, can you combine that with excised tissue, and then using some preclinical models, can we show that multiple methodologies have the ability to demonstrate equivalence? So why is ocular delivery so complicated? Because you have numerous barriers, and therefore the residence time of a drug when administered topical is not very long to begin with. In addition to that, there are multiple pathways for penetration. So the pathways can be either through the tears or the precorneal areas. So all of this becomes very complicated to investigate in a human situation because it is hard to find a representative compartment in which you can measure drug substance. And also it is very invasive because this is the eye. Plasma concentrations are not very representative, and therefore there is a need to come up with a better way to assess performance of an ocular formulation product. A simple test would be to use a cell line. In this case, this is the MDCK cell line. So the idea is very similar to KCO2, and this is essentially showing that for compounds that belong to this fluoroquinolone series, cell permeability is not that different. In fact, it has good correlation with rabbit corneal permeability. Another way to look at this data is where I had the compounds listed. There's correlation to log D. MDCK permeability is in line with what you see with rabbit corneal permeability. So when we saw that this data was appearing to be in the right direction, one of the things that we drew upon was our experience with BCS, where if you remember this slide that I had showed you before, we had maybe 50, 60 compounds, and then we essentially showed that human fraction absorbed is, the KCO2 is predictive of the human situation, the human fraction absorbed. So essentially we took a similar route and said if we were to now evaluate the rabbit corneal permeability model and looked at many drug substances which are used for ocular indications, and these could be for these could be antibiotics, these could be beta blockers, these could be prostaglandins, these could be carbonic anhydrase inhibitors, and then essentially looked at two different tissue barriers, one being the cornea, the second being the conjunctiva, to see which of these is better predictive of what is already known about these markers in the human situation. So we ran about 20 drug substances to come up with a similar type of correlation. Again, from what we had just talked about, it is very critical to get a sense of the variability. Atenolol is a marker for the lower end, for something that does not permeate as well. Antiparin is a marker for the higher end. And as expected, atenolol, which is a lower number, has slightly higher variability than a marker that has better or higher permeability. So anytime you want to use such models as a surrogate, it is important to know that your model has the ability to discriminate. So here is the corneal tissue permeability, literature values which have been performed in other lab settings. So you can clearly see that the compounds you're able to bin, there's a low and a high, as in it can show a difference when a difference does exist, as long as your study is powered correctly. This is the translatability component, where you have the rabbit corneal model versus a pharmacokinetic study performed in rabbit, where you're dosing rabbits topical and looking at accumulation in the aqueous humor. So rabbit cornea, the rate of transfer across a rabbit cornea is representative, can speak towards accumulation in the aqueous humor. And also this is looking at the barrier properties. If these are retained between the rabbit and the human situation, rabbit corneal tissue permeability versus human corneal tissue permeability also shows that we have reasonable correlation. To continue with translation, these barriers can also express certain enzymes and transporters and so on. And this is looking at esterase activity across tissues using a prodrug, dexamethasone acetate, which is converted to dexamethasone base as it transits through the tissue. And the esterase activity is higher in both the Dutch belted and the New Zealand rabbit, because there is no prodrug detected on the other side in either of the rabbit model, but you still have some prodrug that makes it through with the human cornea. So now, knowing that we have a model that can discriminate, that appears to be reasonably predictive of the human situation, we have started using for applications such as formulation comparison. Can we look at flux across excised rabbit corneal tissue to say, well, similarity to a reference to optimize the formulations? These are punctal plugs that were run using this model, and formulation 1 appears to be closer to the reference than the other 3 formulations. Similarly, you could also perform a more involved pharmacokinetic study. It is possible in a preclinical species because the animals can be sacrificed, and you could actually measure drug in all these different compartments, and that gives you a better sense of accumulation and maybe more assurance of equivalence, in addition to the rate of transfer across a certain membrane. The last section of the presentation talks about the use of non-clinical disease model. Therapeutic endpoints. Again, in the ophthalmic space, we have established models for all of these different indications: wound healing, glaucoma, choroidal neovascularization, conjunctivitis, uveitis, and so on. But the model I'll be discussing some more is the glaucoma model. So different drugs can be used for glaucoma. We spoke about this some as we were discussing the validation of the corneal permeability. These can be prostaglandins, these can be beta blockers, carbonic anhydrase inhibitors. So even though they all have a similar endpoint, as in they reduce the hypertension, they act by slightly different mechanisms. Prostaglandins, they increase the uveoscleral outflow. Carbonic anhydrase inhibitors reduce the aqueous production. Beta blockers kind of do both. They reduce the aqueous production and also increase the outflow. So before considering use of a non-clinical disease model, it is important to know that the drugs for which you're developing a matched formulation, an equivalent formulation, in a sense behave similarly between the human and the preclinical situation. So here is some data from beta blockers, and you're looking at human versus rabbit. Few different beta blockers. Again, the response, the reduction in the pressure, the intraocular pressure, is not that different between the two species. So this applies not only to beta blockers. We've also seen that this type of correlation exists with carbonic anhydrase inhibitors, and that this type of correlation also exists with prostaglandins. Again, as we had seen a couple of slides ago, each of these drug classes act by slightly different mechanisms. So maybe it is fair to say that the rabbit is representative of what you could expect in the human situation, at least for these classes of ocular hypertensive compounds. So then we went on to establish a rabbit ocular hypertensive model. So you take normotensive rabbits, which are acclimated for a certain amount of time, and then induce hypertension by dosing with dexamethasone. So you make them hypertensive, and more sensitive, so that any kind of reduction in pressure can be seen, the percent change can be seen to a greater extent than in the normotensive model. So for 21 days, it is induced with dexamethasone, and then you begin treatments in essentially two groups of rabbits. One group of rabbits is where you're dosing maybe your reference or maybe your multiple formulations, and the second could be your test formulation. So you could either compare formulations or try and use this to demonstrate equivalence to a RLD. And then the treatment regimen begins on day 21. You could try and follow the recommended clinical posology and have different periods at which the drug is administered, which is expected to reduce the intraocular pressure, and then readings along the way. So in this particular study, two different drug products were compared, actually a reference and a test, and we had a lot of data going up to this, including the permeation of the API in the two different formulations was very similar. We ran some PK studies to show that the aqueous humor accumulation was also comparable, and now this was the third set of data that we obtained, where essentially what you're seeing here is the decrease in intraocular pressure 3 hours after the drug is administered. And the similarity here between the reference and test shows that here we have yet another piece of evidence as you're trying to build a case for using a multitude of tools, or the idea of multiple platform testing, to show that they're probably interchangeable. So in summary, just to give you a sense that such tools can exist not only in the two areas that we talked about, which is the ocular and the intraoral, but conceptually you could develop and validate such tools for dermal, for oral, for intranasal, intravaginal, and so on. And this gives you a sense for how we would combine different tools and the outcomes from these different tests to drive such endpoints. And the weight of evidence, essentially, the concept is that each of these has the ability to maybe translate into human outcome. But when you have more than one test that can essentially parallel, mirror each other, then you have much more confidence in the directionality of your overall finding, and that can be used for things such as characterization-based equivalence of complex drug products, for IV/IVC, and also for accelerated bioequivalence. And that was my last slide. Thank you very much.
Any questions?
I'm curious about this company.
Yes.
Absorption system.
Yes.
Do you develop your own products, or do you perform a service?
We develop tools, and most times we run services or develop products for other companies.
So what's the future of this activity?
The future is that there is a dearth of tools, translatable tools, in the complex drug product space. So there's a huge impetus for testing. And we hope to be one of the harbingers for bringing about such translatable outcomes and make them available for consumption.
Any other questions? Yes?
I just wanted to put, what's the, not in our source.
That's all right.
What's the regulatory perspective on the, you know, the dermal or not topical
Right.
Delivery, or the dosage formulation that
Mm-hmm.
need to be investigated for the bioequivalence or the
Right. No, that is a very good question. So unlike the oral space, where you have the BCS framework that applies to any drug substance that meets that criteria, currently the regulatory thinking is that you could maybe use these models on a case-by-case basis. So they essentially look at 3 different criteria. First is there a need for what they call an impact product? If there is no generic in the market, then you're beginning to think that, well, maybe the pathway that is currently recommended is not possible to follow, that it has unique regulatory issues. Second is the formulation that you have, can you characterize its physical properties using these types of tests? And if that is a possibility, then they're more open to allowing these pathways. And the third is, how well have you validated this approach? Have you shown its ability to be sensitive, that it can discriminate, that it can demonstrate reproducibility in a consistent manner? And if you can show that by using more than one technique, then the confidence is even more. And there's two ways companies usually approach this. You could either send them a controlled correspondence with some data and say, well, what do you think? The other is where you actually take this pathway with all the data that you believe will get you a positive response and hope for the best. Yes.
I just like to ask for one example. Right now, for the ocular drugs, the stasis. So right now, FDA has some recommendation, and also, so what do you feel that using all these in vitro study or ex vivo study will help you to establish bioequivalence?
It might actually be a lot easier for restasis because there is already a guidance. And as long as you have your Q1, Q2, and Q3, which is prescribed, release from the formulation may be sufficient in this case. I don't think you need to combine that with any type of distribution or efficacy, just because there is a recommendation in place. And the agency has the belief that the formulation can be sufficiently characterized using known PhysChem tools. There was a recent publication from Mansoor's group at the FDA, and they showed how, okay, these are the variables and these are the readouts, and they very nicely described what does and does not impact the formulation. So restasis, I think, is little easier because the FDA has bought into the concept of alternative methodologies for that specific formulation. Yes.
My question is with respect to the regulatory jurisdiction. FDA, we know, is quite open with,
Right.
and take case study, case by case.
Sure.
What's your experience with the differences, or how acceptable would an in vitro method like this be in the EMA?
It's almost like, from what I have seen, the EMA is even more progressive. So I'll give you a couple of examples. FDA is BCS Class 1. EMA is Class 1 and Class 3. We are actually performing a study for a company in Greece, where I think they have some understanding that you could essentially run a rabbit non-clinical study, much like you would run a human clinical study. Now, if you had to run a human clinical study for an ocular product, this has to be in people who have cataracts, because only then during the surgery can you tap into the aqueous humor. You cannot take healthy volunteers and ask for some aqueous humor. But they're essentially running a rabbit study with a multitude of different humors and compartments to show equivalence of a reference to a test. I have never seen a rabbit study run with so much rigor, because they're pursuing a regulatory equivalence endpoint. One more thing that happened even more recently, this was in the dermal space for a corticosteroid. Change in excipient, and EMA said you could either go back and run the skin blanching, which was your basis for original approval, or run a skin permeability study. And the sponsor was surprised because they did not expect the EMA to say skin permeability. The question to us was, what should we run? Well, we cannot run the vasoconstriction. We can run the skin permeability, so you would think I would say skin permeability. But it is an insufficient answer because the FDA is not yet ready for skin permeability by itself. Vasoconstriction actually has applicability in both places. So at least in these three instances that I can think of, we find the EMA is equally progressive. They're just harder to communicate with. Yes, so a lot of people, a lot of languages.